Hello, today we are going to discuss about fundamentals of control of microorganisms in foods. Under this chapter, we are going to discuss about use of high temperatures, use of low temperatures, dehydration, freezing, freeze drying, use of irradiation and use of preservatives. First, we will discuss about use of high temperatures. Exposure to heat denatures proteins, leading to inactivation of enzymes required for metabolism. The heat treatment necessary to kill microorganisms or their spores varies with the kind of organism, the physiological state and the environment during heating. The heat treatment selected depends on kind of organisms to be killed, other preservative methods to be employed and the effect of heat on food. Certain factors affect the heat resistance of cells or spores, like initial concentration of cells or spores in food to be heated, previous history of cells or spores, that is the conditions under which they have been grown, like the culture medium, the temperature, the growth phase and desiccation, etc. There are certain factors which will affect growth of microorganisms. Culture medium components influences heat resistance of microorganisms. Glucose increases heat resistance. Likewise, phosphate and magnesium ions reduce heat resistance in bacteria. Temperature of incubation influences heat resistance of microorganisms. Resistance increases if the cells are incubated at optimum temperature of growth. Growth phase of bacteria has influence on their sensitivity to heat. Bacterial cells are resistant to heat during late log and stationary phases. They are most susceptible during log phase. Water content in cells influences their sensitivity to heat. Desiccation makes them to resist high temperatures. Therefore, spores are more resistant to heat than their vegetative cells. Composition of substrate in which cells or spores are heated influences responses of microorganisms to high temperature. Most of the substrate increases effectiveness of heat as moist heat is more effective in killing microorganisms than dry heat. Substrate at neutral pH is less susceptible to heat than that at acidic or alkaline pH. Solutes containing salt and sugar reduce susceptibility of cells or spores to heat. For efficient application of high temperature used for food preservation, heat resistance of microorganisms in terms of thermal death time and thermal death point have to be taken into consideration. What is thermal death time? It is defined as the time taken at a given temperature to kill specified number of microorganisms under defined conditions. Thermal death point. It is the temperature necessary to kill all microorganisms present in a given substance in defined time, say for example in minutes. For efficient application of high temperature for preservation of food, the rate of heat penetration into food should be efficient. This is influenced by material of the container, like material of container make the glass has slower rate of penetration than metal. Consistency of can contents and size and shape of food item also influences the efficiency of heat penetration into the food item. Say for example, larger one takes longer time and smaller one takes shorter time. Size and shape of contents. Food items could retain their identity, lose their form and become viscous. Then initial temperature of food. Lower the initial temperature, longer time of exposure and higher the initial temperature, shorter the time of exposure required for treatment. Prior treatments, if any, like asepsis, removal of microorganisms etc. affect by reducing the bioburden of food items. Heat treatments employed in food processing. The temperature and time used in heat processing of food will depend on what effect heat has on the food and what other preservation methods are to be employed. Some food such as milk and peas can be heated to only a limited extent without undesirable changes in appearance or loss in palatability, whereas others like corn or pumpkin can undergo a more rigorous heat treatment without marked change. The greater the heat treatment, the more microorganisms will be killed. As a result, heating will produce sterility of the product. The various degrees of heating used in food can be classified as pasteurization, heating above 100 degree centigrade. First, we will discuss about pasteurization. Pasteurization is a heat treatment that kills part but not all the microorganisms present and usually involves the application of temperatures below 100 degrees centigrade. The heating may be by means of steam, hot water, dry heat or electric currents. The products are cooled promptly after the heat treatment. Pasteurization is used under the conditions like when more rigorous heat treatments might harm the quality of product, when the aim is to kill pathogens like in market milk, and when the main spoilage organisms are not very heat resistant such as yeast in fruit juices, when any surviving spoilage organisms will be taken care of by additional preservative methods to be employed as in the chilling of market milk, and when competing organisms are to be killed allowing a desired fermentation usually by added starter organisms as in cheese making. Methods of preservation used to supplement pasteurization include refrigeration, say for example for milk, and keeping out microorganisms usually by packaging the product in a sealed container as in case of fruit juices, maintenance of anaerobic conditions as in evacuated sealed containers, addition of high concentrations of sugars in sweetened condensed milk, and presence or addition of chemical preservatives like organic acids on pickles. Time and temperatures used in the pasteurizing process depends on the method employed and the product treated. The high temperature short time method employs a comparatively high temperature for short time, whereas the low temperature holding for long time method uses a lower temperature for a longer time. The minimal heat treatment of market milk is at 72 degrees centigrade for 16 seconds like in HTST method, and at 137.8 degrees centigrade for at least 2 seconds in the ultra pasteurized method, that is UTH method. Then we will discuss about heating at about 100 degree centigrade. A temperature of approximately 100 degrees centigrade is obtained by boiling a liquid food, by immersion of the container of food in boiling water, or by exposure to following steam. Some very acid food, example sauerkraut, which is a milk product, may be preheated to a temperature somewhat below 100 degrees centigrade, packaged hot and not further heat processed. Blanching fresh vegetables before freezing or drying involves heating briefly at about 100 degrees centigrade. During baking, the internal temperature of bread, cake or other bakery products approaches but never reaches 100 degrees centigrade as long as moisture is present, although the oven is much hotter. The temperature of unsealed canned foods heated in the oven cannot exceed the boiling temperature of the liquid present. Simmering is incipient or gentle boiling with the temperature reaching about 100 degrees centigrade. Frying gets the outside of the food very hot, but the center ordinarily does not reach 100 degrees centigrade. Then heating at above 100 degrees centigrade. Temperatures above 100 degrees centigrade usually are obtained by means of steam under pressure in steam pressure sterilizers like autoclaves. Milk can be heated to temperatures up to 150 degrees centigrade by use of steam injection or steam infusion followed by flash evaporation of the condensed steam and rapid cooling. Then the next process is canning. Canning is defined as the preservation of food in sealed containers and usually implies heat treatment as the principal factor in the prevention of spoilage. Most canning is in tin cans, which are made of tin coated steel or high glass containers. Use of containers that are partially or wholly aluminum or plastics as pouches or solid containers are of a composite of materials. Therefore, the word canning is a general term and it's often replaced by hermetically sealed containers. The heat processes necessary for the preservation of canned food depend on the factors that influence the heat resistance of the most resistant spoilage organism and those which affect the heat penetration. The heating ordinarily is done in retorts with or without steam pressure as the food demands. HTST heat processes now used for some fluid food require special equipment for sterilizing the food in bulk, sterilizing the containers at lids and filling and sealing the sterile containers under aseptic conditions. Other ways of heating cans are by means of a direct gas flame, by steam injection, by heating in a fluidized bed in case of granular solids, and by the hydrostatic sterilizer. Heat is also being combined with other preservative agents like antibiotics, irradiation or chemicals such as hydrogen peroxide for improving preservation. Then we will discuss about use of low temperatures in controlling of microorganisms. Low temperatures are used to retard chemical reactions and action of food enzymes and also to slow down or stop the growth and activity of microorganisms in food. The lower the temperature, the slower will be chemical reactions, enzyme action and microbial growth. A low enough temperature will prevent the growth of any mesophilic or thermophilic microorganisms. As the temperature drops from its optimal temperature toward the minimal, the rate of growth of the microorganisms decreases and is slowest at the minimal temperature. Cooler temperatures will prevent growth of an organism, but slow metabolic activity may continue. Therefore, the cooling of food from ordinary temperatures has a different effect on various organisms present. In general, freezing prevents the growth of most foodborne microorganisms and refrigeration temperatures slows down the growth rates. Commercial refrigeration temperatures that is lower than 5 to 7.2 degrees centigrade effectively retard the growth of many foodborne pathogens. Common or cellar storage. The temperature in common or cellar storage usually is not much below that of the outside air and seldom is lower than 15 degrees centigrade. Root crops, potatoes, cabbage, apples and similar food can be stored at these temperatures for limited periods. Then we will discuss about chilling or cold storage. Chilling storage is at temperatures not far above freezing and usually involves cooling by ice or by mechanical refrigeration. Most perishable food including eggs, dairy products, meat, seafood, vegetables and fruits may be held in chilling storage, commonly used deep freezers for a limited time with little change from their original condition. Factors to be considered in connection with chilling storage include the temperature of chilling, the relative humidity, air velocity and composition of the atmosphere in the storeroom, and the possible use of ultraviolet rays or other radiations. Now we will discuss about cooling temperature. The lower the temperature of storage, the greater the cost will be. Therefore, although most food will be kept best at a temperature just above their freezing point, they are not necessarily stored at this low temperature. The chilling temperature is selected based on the kind of food, the time and conditions of storage. Some varieties of apples undergo low temperature breakdown at temperatures near freezing. Then the next method of control of microorganism is dehydration. Dehydration is defined as the application of heat under controlled conditions to remove the majority of the water normally present in a food by evaporation. Dehydration is intended to halt or slow the growth of microorganisms and the rate of chemical reactions. The removal of water provides food processors excellent opportunities to reduce volume and weight, extend shelf life and convert liquids to powdery products such as instant coffee or a vegetable soup base mix. Food dehydration refers to the nearly complete removal of water from foods under controlled conditions that cause minimum or ideally no other changes in the food properties. Such foods, depending on the item, commonly are dried to final moisture within the range of about 1 to 5%. Examples are dried milk and eggs, potato flakes, instant coffee and orange juice crystals. Such products will have storage stability at room temperature of a year or longer. A major criterion of the quality of dehydrated foods is that when reconstituted by the addition of water, they may be very close to or virtually distinguishable from the original food material used in their preparation. In food dehydration, the technological challenge is especially difficult since very low moisture levels for maximum product stability are not easily obtained with minimum change to food materials. Whatever method of drying is employed, food dehydration involves getting heat into the product and getting out moisture. The factors important in this regard to be considered are surface area, time, temperature, evaporation, air velocity, humidity, atmospheric pressure and vacuum. Freezing. If you take freezing, the storage of food in the frozen condition has been an important preservative method for centuries where outdoor freezing temperatures were available. With the development of mechanical refrigeration and the quick freezing processes, the frozen food industry has expanded rapidly. Even in the home, the freezing of food has become extensive now that home deep freezers are readily available. The lower the storage temperature, the slower will be any chemical or enzymatic reaction, but most of them will continue slowly at any temperature now used in storage. To avoid this as a common practice, the enzymes in vegetables are inactivated by scalding or blanching processes. The efficiency of freezing as a method of preservation depends on various factors like selection and preparation of food, method of freezing, conditions of food and its method of harvest, etc. Selection and preparation of food for freezing. The quality of the food to be frozen is of prime importance. Fruits and vegetables are selected on the basis of their suitability for freezing and their maturity and are washed, trimmed, cut or otherwise pre-treated as desired. Many foods are packaged before freezing. The rate of freezing of food depends on a number of factors such as the method employed, the temperature, circulation of air or refrigerant, size and shape of package and kind of food. Sharp freezing or slow freezing usually refers to freezing in air with only natural air circulation or at least with electric fans. The temperature is usually minus 23.3 degrees centigrade or lower, but may vary from minus 50 to 29 degrees centigrade, and freezing may take from 3 to 72 hours. Then coming to quick freezing. Quick freezing implies a freezing time 30 minutes or less and usually involves the freezing of small packages or units of food. Quick freezing is done by three general methods. One is direct immersion, second one is indirect contact, and third one is air blast freezing. Direct immersion of the food or the packaged food in a refrigerant. Second one is indirect contact with the refrigerant where the food or package is in contact with the passage through which the refrigerant at minus 17.8 to minus 45.6 degrees centigrade flows. The third one is air blast freezing where air is blown across the materials being frozen. Certain fruits and vegetables, fish, shrimp and mushrooms now are being frozen by means of liquid nitrogen at minus 196 degrees centigrade. For dehydrofreezing, fruits and vegetables have about half their moisture removed before freezing. Now we will discuss about response of microorganisms to freezing. Various factors influence microorganisms variably during low temperature preservation. Some microorganisms die, some are injured and some are not damaged. The factors that influence are the kind of microorganism and its state, the freezing rate, the freezing temperature, the time of frozen storage, the kind of food and alternate freezing and thawing. Freeze drying is the process of dehydrating frozen foods under a vacuum, so the moisture content changes directly from a solid state to a gas state without undergoing sublimation state. In this process, the product maintains its original size and shape with a minimum of cell rupture. Now we will discuss about the steps in the process of freeze drying. The fundamental steps are freezing, vacuum, heating and condensation. The first step, freezing. Here in this step the product is frozen. This provides a necessary condition for low temperature drying. Second step is vacuum. After freezing, the product is placed under vacuum. This enables the frozen solvent in the product to vaporize without passing through the liquid phase, a process known as sublimation. Third step is heating. Heat is applied to the frozen product to accelerate sublimation. Then the fourth step is condensation. Low temperature condenser plates remove the vaporized solvent from the vacuum chamber by converting it back to a solid. This completes the separation process. There are several benefits of freeze drying that we will discuss now. Freeze drying retains original characteristics of the product including its color, form, size, taste, texture and nutrients. Reconstitutes to original state when placed in water. Shelf stable at room temperature and cold storage not required. The weight of the freeze dried product is reduced by 70 to 90% with no change in volume. The product is lightweight and easy to handle. Shipping costs are reduced because of the light weight and lack of refrigeration. Low water activity virtually eliminates microbiological concerns. Offers highest quality in dry product compared to other drying methods. Virtually any type of food or ingredient, whether solid or liquid, can be freeze dried. Then the next method is irradiation. Food irradiation is the process of exposing food to ionizing radiation to destroy and check the multiplication of microorganisms, bacteria, viruses or insects that might be present in the food. Further applications of irradiation include sprout inhibition, delay of ripening, and improvement of rehydration. How is food irradiated? Bulk or packaged food passes through a radiation chamber on a conveyor belt here. The food does not come into contact with radioactive materials, but instead passes through a radiation beam like a large flashlight. The type of food and the specific purpose of the radiation determine the amount of radiation or dose necessary to process a particular product. The speed of the belt helps control the radiation dose delivered to the food by controlling the exposure time. The actual dose is measured by dosimeters within the food containers. Usually cobalt 60 is the most commonly used radionuclide for food irradiation. Irradiation is most useful in three areas, basically in food preservation, food sterilization, and control of food pathogens. Then the next method is use of various preservatives in food preservation. The ideal antimicrobial preservatives are organic acids and their salts, nitrites and nitrates, sulfur dioxide and sulfides, ethylene and propylene oxide, sugar and salt, alcohol, formaldehyde, wood smoke, spices and other condiments, other groupings of chemical agents, antibiotics, and developed preservatives. We will discuss all these things one by one now. All these chemical preservatives and antimicrobial preservatives generally we will use to preserve the foods. Antibiotics. Aureomycin and terramycin inhibit protein synthesis. These are two antibiotics. And another important antibiotic is nisin, which is used in cheese to suppress the anaerobes. Natamycin is also effective against yeast and molds. Natamycin is used in orange juice, fresh fruits, sausages, and cheese. FDA approved the use of chlorotetracycline and oxytetracycline tape for preserving poultry. Then there are several developed food preservatives that will be used in preservation of various foods. Food fermentations may serve either or both of two purposes. One is to produce new and desired flavors, and the second one is to help preserve the food, like fermented meals, sauerkraut, and other milk based food products, that is fermented milk based food products. The preservatives produced in foods by microbial action are for the most pot acids and alcohol. The preservative effect of these substances nearly always is supplemented by one or more additional preservative agents such as low temperature, heat, anaerobic conditions, sodium chloride, sugar, air, or added acid. Today we have learned about fundamentals of control of microorganisms in foods by various methods like use of high temperatures, use of low temperatures, dehydration, freezing, freeze drying, irradiation, and use of preservatives, especially antibiotics, and developed preservatives in food preservation. Thank you.